introduction about BK virus. I'm sure some of you have come because you already know things about BK virus, but for those who might be completely new to the topic, BK is thought to be the initials of the first patient BK virus was described in, but that's actually disputed and it's not completely clear how it came to have that name. It's a virus that most people already have. The vast majority of people have it by the age of 4 years. If your immune system's working okay, it doesn't cause any harm, and it simply lives there in your kidneys and you never know about it. But the issue occurs when your immune system's reduced, and in the case of a kidney transplant patient, this is by your anti-rejection medicines, as I'm sure you know, also called immunosuppression. So how does BK virus potentially at least affect kidney transplants? Well, it can worsen transplant function because the immunosuppression drugs mean that the immune system doesn't keep it in check in the way that it does in someone who doesn't have to take those medications. In the worst case scenario, it can even cause a transplant to fail. The only proven option we have to help with BK virus is reducing immunosuppression, though doctors do try other medications as well, and I'm sure some of the patients here may be aware of that. However, reducing immunosuppression runs the risk of causing rejection and thus shortening the transplant's life. So the presentations this evening are to bring you up to date about research in the UK, which is really trying to do two things: trying to get a better understanding of the virus and how it affects our patients, and trying to find new ways to combat it. Before we get to our presentations, Kidney Research UK have very kindly teamed up with one of our patients in order to provide us with a short video. It's about 4 minutes, telling us about the patient experience of BK. So we're going to start with that and move to the presentations afterwards. I'm going to play that for you now. Any problem with the sound, please let me know.
Hi, my name is Fez. I have been a renal patient all my life. I have used all modalities of dialysis within my lifetime, and I am currently on my third transplant, which I had in July 2020. Before my transplant, I knew nothing about the BK virus, and before the transplant, I wasn't really told anything about the BK virus either. So it was approximately 6 to 9 months after my transplant. I started to feel a bit of pain whilst urinating, and I was getting tummy aches. Originally, I thought it was just a UTI, so urinary tract infection, because I've had lots of them, and it felt very similar to that. I went in to have bloods done, and my creatinine levels had risen, and my GFR had come down somewhat. Being newly transplanted, they were a little bit worried, so they admitted me and kept an eye on me. They ruled out UTI, and so they did a biopsy, and they found out that I had BK virus. So initially, I was basically told by my consultant at the time there was no specific treatment for BK virus. It was more of a balancing act of trying to reduce my immune suppressants, to try to get rid of it, but not reduce it too much that then my transplanted kidney gets damaged. So some adjustments were made with my immune suppressant drugs. We found a level that kind of worked where it wasn't damaging my kidney any more than needed, but also the BK virus levels in my blood were slowly decreasing. So I'm not still officially cleared. So it's been about, I don't know, it's been about 2 years approximately. But where it was at a very higher level before at the start, it's decreased to a much smaller amount in my blood system. So yeah, it's not completely gone, but there is like a small amount, trace amounts in my blood system still. I don't know if it will ever fully disappear, but as long as it's not causing any damage to my transplant and I'm able to take my medications as I need, I've been told that it should be okay, and they'll always check in my bloods if it does increase again, but it seems to be at a very minimal level now. So as I mentioned earlier, I thought it was a UTI, and because I've had quite a few UTIs in my lifetime, I wasn't that worried. But then when they told me it was a new virus I hadn't really heard of, and there was no specific treatment, and they had to mess with my immune suppressant drugs, I was quite worried then. Being my third transplant, it's not something obviously I want to lose, and have to go through all the rigmarole of going back on the transplant list, etc. So at that time, I was very worried indeed. Every blood test that I was going for afterwards, I wanted to know what the BK level was in my blood. Was there any improvement? Was there any not improvement? So yeah, at first it was very, very stressful, and it played on my mind quite a lot. So currently, I think I'm a bit more relaxed about it. I've been told that it's not at the very high levels that it was at, and it has decreased to a very, very minimal level, where it shouldn't affect my kidneys or my health in general, for passing urine, etc. So I'm not as worried as I was, and I think because I'm slightly more comfortable going out or going to the hospital when needed, I'm always kind of assured that I can go and get a blood test if I need to. And again, they regularly check it when I have my blood tests. So unless there's a spike or I'm feeling unwell or there's a problem with my urination, generally I feel a lot calmer than I did.
So many thanks to Fez and to Kidney Research UK for providing that for us. I'm just going to stop sharing my screen now so that we can move to our first presenter. So Andrew, if you'd like to get your slides set up, and I'll just introduce you. So Andrew McDonald is our first presenter today. Andrew is Professor of Tumour Virology at the School of Molecular and Cellular Biology in the University of Leeds. He's funded by both Medical Research Council and Kidney Research UK to understand how viruses of the type that BK is cause disease. Thank you very much for speaking for us today, Andrew. Please go ahead when you're ready.
Absolute pleasure. Matt, can you see my slide?
We can. Thank you very much.
Excellent. And can you hear me okay?
Yep, that's great.
That's great. So it's a real pleasure to be here tonight to tell you about some of our research on BK. So I've been very fortunate that Kidney Research UK has funded work in my lab on BK virus for about the last 10 years or so. And really, I suppose what I want to talk to you about today is some of the work that we're doing trying to understand how this virus gets into a cell. So like all viruses, BK is a parasite. It cannot reproduce and make copies of itself unless it can get inside a cell. So it has to find a way of attaching to a kidney cell and of getting inside that cell. And the surface of all cells, including kidney cells, is completely covered with almost like a canopy of proteins and other molecules that reach out from the inside of the cell to the exterior and allow that cell to be able to communicate with its surroundings. And in order to gain entry to cells, BK needs to find a particular protein to bind to, and it's almost, you can think of this almost as a lock and key effect. The virus is the key and it has to find a lock on the surface of the cell. It interacts with that lock and it uses it to gain entry to the kidney cell in order to reproduce itself. And it's this reproduction of the virus that causes damage to the kidney cell and causes the pathology that you will all be aware of. So in order to study BK virus, we decided to take an approach to try and visualize what this virus looks like at very, very high resolution that would enable us to try and generate drugs targeting that lock and key effect. And so we're fortunate that we can grow this virus in the lab in cell culture. Here you're seeing an electron micrograph of the virus. So the little circles that you can see here, these are actual BK viruses that we have grown and purified in the lab. And then we take this virus and we put it in a specialized facility that we have at Leeds called a cryo-electron microscope. So the microscope is this very large obelisk here. It's about 2 or 3 meters tall. They're absolutely huge machines. And what this microscope allows us to do is see very small things such as viruses at very, very high resolution. And so we put our virus onto a carbon grid, and I'll see whether the movie will work. We take that grid and then we plunge it into extreme cold very, very quickly. And you'll see it plunge in a moment. There it goes. It gets plunged into liquid ethane. So this is extreme cold temperature. And what that does is it freezes the virus onto the grid. And we then shove that grid into the microscope that I showed you earlier. And we look down the microscope at our virus. And this, ladies and gentlemen, this is BK virus. So this is the virus that causes all the trouble that we're going to hear about in tonight's session. And what the electron microscope allows us to do is look at this virus in extreme detail. And what we're zooming in on here are these pentamers. So this is the virus key that it uses to bind to that kidney cell onto the lock and to get inside the cell to cause infection and to cause disease. And because this microscope allows us to see these viruses at such high resolution, we can use several techniques in the lab to take this structure that we're visualizing here of this key, and we can start to try and play with the key to see whether we can identify drug molecules that might prevent the key getting into the lock and thus prevent BK from getting inside a kidney cell. So we take that structure and we have libraries of the host proteins that bind the locks, and we can determine the structure of the key bound to the lock. So these sort of knob-like protrusions, these are the protein that the virus uses to get inside the cell. And the little purplish blobs that you see on there, they're the host protein, this protein here that sits on the surface of the kidney cell. And they are bound to the virus proteins. And that is the key binding to the lock. It will turn and it will allow the virus to gain entry into the cell. So we can take our knowledge of that key interaction and we can screen very large libraries of drug molecules that we have in a computer. So this is all done in a computer. Basically, the computer looks for drugs that might be able to squeeze in between that lock and key and block the two proteins from binding to each other, and therefore stop the virus from being able to infect a cell. And so we take our best compounds from the computer and then we screen them. This takes a very long time. We screen them in the lab to see whether they will block infection. And occasionally we get a hit. One of these drug-like compounds will prevent the virus from being able to infect kidney cells. And what we're doing at the moment is we're taking these promising compounds and we're seeing whether we can make them better, seeing whether we can change little bits of them that make them more effective at blocking that lock and key interaction. So one of the things we're doing at the moment that we're very excited about is all of the work with that microscope is using virus that we've grown in the lab that we're looking at almost in a test tube. But what's really important is to try and understand what's happening to BK virus when it gets inside a cell, because we really do not understand in great detail how this virus travels through the cell, how it interacts with the cell, and how it causes the disease. And a better understanding of that may enable us to develop better therapies. And that is where we're using an even more up-to-date form of microscopy called tomography. And basically what this does is it takes that fantastic microscope that I told you about, and it allows you to look inside a cell using that microscope. So rather than look at an isolated virus, we're actually going to look inside an infected kidney cell. And so what I'm showing you here with the little white lines, these are BK virus just there. And just here, you can see a load of them packed together. These are viruses inside an infected kidney cell. And using that microscope, we're going to be able to look at them in such high resolution as I showed you earlier. And it will reveal brand new knowledge and information about this virus, but in the context of an infection rather than in a test tube. And we're really excited about this. This is cutting edge in terms of structural biology. So what I've told you today is that this electron microscope allows us to see the virus up close, both in a test tube, but also inside a living kidney cell. And we can use this information to better understand how the virus infects cells. And hopefully, from that information, we can begin to design drug candidates to prevent infection. So I just want to finish by thanking the two people in my lab who did that work, who were both PhD students at the time, Karina, who was funded by Kidney Research UK, and Dan, who was funded by the Wellcome Trust. And I will end it there, and I will thank you for listening. And I'm happy to answer questions at the end.
Thank you very much indeed, Andrew. That was great as our first talk and a good introduction to some of the other things we're going to move on to. Our second presentation today is going to be from Simon Baker. Simon, would you like to start sharing your slides so you're set up, and I'll just introduce you. Simon is a Kidney Research UK Intermediate Fellow who's based at the University of York. What Simon's work seeks to understand is why kidney transplant recipients appear to have a 3 times greater risk of bladder cancer than the rest of the population. Is that something to do with the BK virus itself? So thank you very much for being here to talk to us today, Simon. I'll hand over to you.
Thank you, Matt, and thank you everyone for joining us this evening. Yeah, so I work at the University of York, and as Matt says, my work seeks to understand if BK virus could be a risk factor for bladder cancer. And yeah, the reason that we're focusing on this is because kidney transplant recipients are at a 3 times greater risk of bladder cancer. And at the moment, we don't really understand why. When we talk about bladder cancer, people generally agree that the main risk factor for bladder cancer is smoking. And if you look on the Cancer Research UK website, for example, most of the information you'll find there is about how smoking is the main risk factor for bladder cancer. But actually, when we look in the bladder cancers that form in patients, the data that we find in the DNA there doesn't fit smoking as the main risk factor for bladder cancer. So we've been looking at BK virus, and from Matt's introduction, I don't need to spend much time explaining to you that BK virus infects nearly all of us in childhood. It then goes into a kind of persistent dormant phase in our kidneys during adulthood, but that during adulthood, if we become immune limited, and in kidney recipients that's related to immunosuppression, but actually also in all of us as we age, we can find BK virus in the urine. And it's that BK virus in the urine that we think could be causing bladder cancer. So my research is based on taking pieces of human bladder and ureter tissue, and you can see that here. The dotted line, the cells that are above that, you can see here. This is the epithelium that lines the ureter, so the pipe that goes from the kidney into the bladder, and also the bladder. And these epithelial tissues, they form all of the barriers around your body. So things like your skin, the lining of your guts and your lungs, but also the lining of your bladder. And this is the barrier that keeps the urine in the bladder and stops you from reabsorbing the toxins that your body's trying to get rid of. What we do in the lab is we separate off this epithelial tissue and leave the tissue that's beneath, and we use it for other things. We take the epithelial cells and we expand them in these kind of special flasks in culture. So we can, from a very tiny biopsy, we can get huge numbers of cells from a healthy patient. Most of our cells actually come from kidney donors, because what happens is that the surgeon taking the donated kidney takes almost all of the ureter from the donor, but then plums very little of the ureter into the recipient. And so normally that ureteric tissue would be waste, but actually we take it into the lab and use it in our research. And what we can do is, after we've expanded these cells in numbers, we can use special methods to turn the tissue back into this multi-layered tissue again that forms a barrier and has all of the features just like it did in the body. And then we can upset it by infecting it with BK virus and see what the effects are and study how BK virus influences the biology of the bladder. And the main thing that we do is to look for patterns in damage of the DNA. We look at DNA damage in cancers, but we also look at DNA damage in our laboratory models, and we try to compare them. And the reason that we do that is that when we look across the whole genome and try to capture every mutation that forms in the genome, we start to see patterns, and we think of those more like fingerprints at a crime scene. Those patterns of DNA damage can be used to identify the cause of the damage. And so what I'm going to do is show you some real data from our lab that seeks to help you kind of understand the way that we approach this. So whose fingerprints do we find on the DNA of bladder cancers? Well, we use these graphs, and they can look intimidating because they are big graphs and they have lots of bars in them, but you don't need to worry about them. I always tell everyone that I'm showing these mutational signatures to, that all we're looking at is patterns. So keep thinking about them as fingerprints and see the patterns. So this is the mutational signature that we see in almost every bladder cancer. So more than 9 out of 10 bladder cancers have this pattern of damage in them. And the Sanger Institute analyzed the bladders of aging people who didn't have cancer. And this is the pattern of damage that they saw. And you can see that the pattern is quite different. The peaks are forming in different areas. We did a study in 2020 where we took our bladder cells and we put smoke toxins onto them, and we looked at the DNA damage that was caused by smoke toxins. And this is the pattern that we see forming. You can see the light blue bars coming up here. And these light blue bars, this pattern here, is very characteristic of lung cancers. We see this pattern of damage in nearly all lung cancers. But importantly, we don't see this pattern in bladder cancers. But then we moved on more recently, and this data we haven't actually published yet, so this is very cutting edge. We do our BK virus infections in the laboratory, and we see this pattern of damage. And so hopefully what you can see here is a similar pattern forming to what we saw in bladder cancers. We're getting this same repeating pattern of spikes in the red section, and also to a lesser extent in the black section here. And so we can start to match up these fingerprints that we see in bladder cancer with those that we're seeing in lab infections of BK. And the reason that we can join these up is that the BK virus infections that we do in the lab induce a family of enzymes called apobecs. And what is an apobec? Well, it's part of your host defense against viruses. Apobec enzymes destroy viral genomes, but unfortunately, sometimes whilst they're in the process of attacking the DNA from the virus, they also damage our own host DNA, our DNA of our own cells. And so that's what we think is happening here as kind of part of the cell's approach to try and limit the virus. We're also damaging our own DNA, and that could be potentially leading to cancer. And the other thing that we're looking at the moment that we're really excited about and we think is quite hopeful for the future is that actually the tissue that lines the bladder can fight infection. So again, here we're looking at the tissue that lines the ureter in the bladder, and this tissue has been infected with BK virus, and the BK virus is labeled in brown. So the blue parts are healthy tissue, and you can see the nucleus of the cell. So where all the DNA is are these blue blobs. And you can't see the edges of cells because they're all well integrated into a tissue that's functioning together. But what you can see is these brown blobs that are being encapsulated first, and this is all the virally infected cells being packaged up together and then ejected out of the top of the tissue and pushed out into the urine. And so what we hope is that in future research, we'll be able to seek to improve this process and enhance it, make the tissue extra sensitive to viral infection so that it can boot out the virus earlier before any of this damage is done to the tissue. So in conclusion, our research suggests that BK is likely to be an important risk factor for bladder cancer. And what we really need now is to get more data from patient samples. And we also think that this is not just a problem for kidney recipients. We think this is a problem for everyone, because as we age, you can see what happens is that we detect more virus in the urine, or we detect it more frequently in the urine. So above the age of 50, about a third of people will have BK virus detectable in their urine. And this kind of is associated with the fact that as we age, our antiviral immunity starts to drop off. We're less good at fighting viral infections. And so this is a really important issue for us to deal with, and we're trying to work on this problem generally. So I'd just like to say thank you to everyone for attending this event and listening. As part of the British Transplantation Society work that we've been doing, we've developed a new patient information leaflet. So if any of the patients on this call are interested in giving us some feedback or contributing ideas towards this patient information leaflet, please do get in touch with me, because I'd be really happy to discuss it. And a big thanks goes to all my collaborators and Kidney Research UK for funding my fellowship and allowing me to do this research. Thank you.
Thank you very much indeed, Simon. Absolutely fascinating. Thank you for sharing all that with us today. I'm going to take the prompt from your suggestion there about people contacting you, just to make people aware that this event this evening is a first step in our patient engagement in the UK-wide BK research. One of the things that we're going to do at the end of this process, a few days after this event, is email everyone who's registered and give you the opportunity to be involved in other ways in the future if you'd like to. And that will include a way that you can contact Simon for what he's just said through us. So we're going to move to our third talk now. Our third speaker is Colin Crump. Colin, feel free to get your slides up ready to go. Colin first studied biochemistry in Bristol at the university there, and he began studying viruses in the USA in a fellowship in Oregon. He actually came back to the UK in 2002 to Cambridge, and it's there that he started his own research group. He's currently a senior lecturer in virology there and will shortly become Professor of Molecular Virology in October this year. Many congratulations, Colin. He's actually been interested in BK virus since 2012, and so he's been working in this area for over a decade. Look forward to what you've got to share with us, Colin.
Thank you, Matt. Just on getting my sharing working. Let's see. Right, can you see my screen?
That's great. We can. Great.
Okay, so thanks very much. It's a pleasure to be here and talk to you about some of the work we've been doing in my lab. What I'm going to tell you about today is some work we've been doing to try and find new ways to prevent BK virus infection. In my slides I call it BK polyomavirus because that's the full name, but the same thing, BK virus. It's just polyomavirus is the family it's part of. So as Matt said, I'm in the Department of Pathology down at the University of Cambridge. So the one thing to keep in mind, as has been mentioned before, viruses can only grow inside our cells, so they're entirely reliant on the processes inside our cells to be able to cause infection. That's true for all viruses. And it's particularly true for BK polyomavirus. As has been mentioned, it's a very small virus, so it's deceptively simple and only expresses a handful of its own proteins. So about 7 proteins is all the virus has of its own. And it does everything it needs to do to replicate and reproduce inside our cells and kidneys with just these 7 proteins. So the virus lives inside the cells of our kidneys, as Matt mentioned at the beginning of the session, and generally causes little or no harm. So most of us have this virus in our kidneys, and it persists silently within our cells. And then every now and then there'll be some sort of event that causes it to wake up, or sometimes called reactivate. And this makes the virus sort of go back into its reproduction cycle, and then new viruses are made, normally in the cells that line the tubules of the kidneys, and then they get excreted in the urine. And then they can go on to infect the bladder cells, as Simon was talking about. And again, as Matt mentioned right at the beginning, it's the immune system that normally helps to control the virus. So when the virus reactivates, the immune system will sense that and then kick into action and control the virus and halt the production of those viruses and the shedding. So then it goes back into sleep, and then every now and again, it'll wake up again. So it's kind of how the virus has adapted to live with us over a long period of evolution. So this really lives in a dynamic equilibrium with us and doesn't really want to cause much harm. So really, diseases caused by this virus are basically caused by excessive reproduction of the virus. So usually when the immune system is suppressed a bit, the virus goes a bit sort of crazy and happy, makes lots of copies of itself, and that's when you can start getting disease. So then what is the best way to prevent this? And as I'm sure you're aware at the moment, we don't really have any good directly acting therapies to treat BK polyomavirus. No antiviral drugs that work on the virus directly that are useful in most situations. So that we kind of, the clinicians have to resort to tailoring the immune suppression to allow the immune system to get better and to help suppress the virus, which has its own complications. So what we do in my lab is study how viruses interact with cells, and we're interested in the sort of basic biology of what viruses do to cells to make them into a good place for that virus to reside and to replicate. And this, in terms of our BK research, what we want to do is find proteins that our cells have that this virus really needs to reproduce and infect the cells. Now, there's lots of really fantastic new techniques in science that have been developed over the last sort of 10, 20 years. One really useful research technique that allows us to assess the importance of every single gene that our cells express for any particular process in so-called genome-wide knockout screens. And in essence, what you do here is you have lots of cells which you have growing in the lab, and then you treat those cells so that in each cell, they lack one of the approximately 20,000 genes that our bodies express. And so then you have this big population where there's lots of cells, each lacking one function of that cell. And then you can treat those cells with whatever you want and see if there's a change in how that cell responds to it. And in our case, we can infect these cells with the BK virus and then see whether or not they can support reproduction of that virus. And so what we're looking for are cells which are basically stop the virus reproducing, and that's because of what's lacking in that cell. So which gene has been knocked out. So we can isolate those cells which don't replicate the virus, and then find out which gene is inactivated. And that will give us a nice list of candidate genes that are maybe needed by the virus to replicate successfully in our cells. Okay, so it's a bit like fishing. So it's a big, very, very expensive, complex fishing exercise to look for things which the virus needs inside our own cells. And this is the sort of data we get out of these type of screens. So this is a big complex graph with lots and lots of dots on it. But essentially, every single dot on this graph represents one gene in our body. And so they're just ranked alphabetically along the x-axis along the bottom, and so then going up on the graph on the y-axis, as it's called, is basically a measure of how important those genes in our body are for the replication of the virus. So the ones up high, up here, are the ones which are particularly important for the virus to reproduce in our cells, in our kidney cells. As you can see, most of them are down the bottom, so the virus doesn't really care about most genes, but there's some genes or proteins in our cells which the virus really cares about. And this gives us a lot of candidates to then go and test and see, okay, why does the virus need these, and is there any way we can use that knowledge to help fight the virus? And there's one here which is highlighted in red, which we've studied, a protein called MAT2A. I won't go into details of it, but basically it's an enzyme in our cells which is important for lots of things to function properly in our cells. So then there comes the question, are there any drugs that can target these proteins we've identified in our cells that the virus needs? So this is where we got really interested in this protein. So this protein here called MAT2A is actually an enzyme which is important for some cancers to survive. And so if you knock out reduce the function of this protein in some cancer cells, the cancers die. And so, as you'd imagine, there's been a lot of interest in pharmaceutical companies to develop drugs which target this enzyme as a potential cancer therapeutic. And actually, there's a few drugs that target this protein that are in so-called phase 1 clinical trials, the really first clinical trials for potential cancer therapeutics at the moment, which is exciting because there are actual drugs available to this protein. And so, yes, there are drugs available to at least one of these candidates we've identified. So do these drugs inhibit BK virus reproduction? So here's just some bit more data. So this is just taking two inhibitors of the same protein. And these graphs show here on the left axis how well the virus reproduces. And this is in cells in culture in the lab. And the blue bars are just in normal cells. And then the colored bars are where we treated the cells with the inhibitors of this protein. And this is just increasing amounts of the inhibitor. So as you get increasing amounts of inhibitor, you get less reproduction of the virus for both inhibitor 1 and inhibitor 2. I haven't bored you with the names of these inhibitors because they wouldn't mean much. So this is really sort of exciting because we've got two independent drugs to this protein we've identified, which at least in the lab can both inhibit the reproduction of the virus. So hang on, why isn't it moving? There we go. So that's really exciting, and there's potential for you developing these inhibitors further for perhaps a therapeutic to treat BK in patients. But are there other targets? So obviously there's lots of proteins here that you can see, or lots of little dots which are coloured, which are high up on the graph, which may be important for the virus. And we've done other screens, and this is just another very complicated slide with lots of names on it. And this is just representing things which happen in the cell. And all these names are things we've identified in these screens that are important for the virus, or may be important for the virus. So we've got lots and lots of candidates that we can follow up. And at least one more of these seems to be important for the virus, and there's a hopeful potential for combination therapy. So this is just another way of representing how we look at the reproduction of the virus in cells in the lab, and treating with two different drugs. So drug to protein number 1 is one of the first ones I showed you before, to that MAT2A protein. And so this is showing the replication of the virus over time. So the black line is no drugs at all, so the virus replicates to this level. With drug to the first protein, you get an inhibition here. And with another drug to a different protein we've identified, you get a similar inhibition. But we can put both drugs in together, and we get an enhanced inhibition of the replication of the virus. So the chance of maybe combination therapy to target more than one mechanism inside our cells to bring down the replication of the virus, to help suppress the virus replication, and then maybe wouldn't need so much immune suppression to control the virus and the diseases caused by it. So what are the next steps? So everything I've told you about, we've done in the lab, in human kidney cells, which you can grow in the lab in culture. Now, these are cells which are grown in plastic dishes, and obviously it isn't quite the same situation as what's in the real, in the body, in the actual kidney itself. So what we need to do is to test drugs in experimental systems that most closely mimic the natural kidney itself, so in a sort of three-dimensional sort of organ-type architecture. And there's this fairly new development in sort of biological science and medical sciences, where there are things called so-called organoids, where it's a way of taking some different types of cells together and growing them together in a three-dimensional sort of way, where they grow into a sort of thing which looks like the real organ and mimic the natural architecture of the tissue that you're looking at. And this can be done for kidneys, at least only for the tubules where the BK virus grows. So what we are planning to do now, and we've just got some really generous funding from the Kidney Research UK to develop this, is to use organoid models in BK infection research to then test the replication of the virus, and then how we can treat it with these different drugs we've identified. And then the ultimate aim of this is to generate lots of data to then go to drug companies who have made some of these drugs to convince them to invest some funds and time to perhaps look into clinical trials to see whether any of these drugs or combinations can actually have a benefit in patients. And just to give you an example of what this looks like, so this is this organoid system which we plan to set up. So we use these very complicated sort of plates, which are with lots of tiny little tubes in them, and you can grow human kidney cells into what basically look like normal, proper kidney tubules that are normally in the kidneys, with all the normal cellular architecture. And this is what we're going to use to infect with the virus, and then treat with these different drugs that we've identified. So, just to sum up, hopefully there is some hope, but it's going to take some time and lots more work to try and work out whether these drugs are good and whether they work, how well they work in patients. And just to show you the people who are involved in this, there's 3 people in this photo: Natalia, Laura, and Sophia have been involved in this work over the years, and it's been great to have funding from the Kidney Research Trust to develop this, as well as other cases. And thank you for your attention, and I look forward to some discussion.
Thank you very much indeed, Colin. That's fantastic. And it's great of you to leave us with a little bit of hope there for potential interventions to try to help here. So we're going to turn now to some of the questions that have been coming through on the chat, and I'm going to take these with Sian, my co-host, and Simon, Colin, and Andrew will all be available to join in on the scientific side. Sian, do you want to take the wheel and begin the question discussion?
Yes, thanks very much, and thank you to all 3 presenters for really excellent summaries of the current state of the art for BK research in the UK. The questions we've had so far have related to clinical management and what the presence of BK viremia means for your overall level of immunosuppression. So I'm just going to give a few thoughts on that, and then I'm sure Matt will also have some as well. One of the most difficult aspects for me looking after patients with BK in the blood, and then patients who have abnormal function of their transplant due to BK, is just how variable it is. So we see patients who have quite a low level that's very easily controlled by reducing the immunosuppression, And then we see patients who have a much more aggressive viral proliferation, which causes significant damage to the transplant quite quickly. And it's often difficult at the beginning to know exactly how the patient is going to progress when you first become aware that they have BK replication. Generally, it is an indication that the immune system is too low. So it's been suppressed to the point where it can't control this virus. And we know other viruses can also cause problems for transplant recipients. So our first line management is to reduce the intensity of immunosuppression. However, as Matt mentioned in the introduction, we're walking a tight line with that, in that we want the part of the immune system that can deal with BK to reconstitute and be able to do that. But we don't want the part of the immune system that can cause rejection to become a problem. So it is quite careful management of the immunosuppression, and we often do this in stages. So we don't immediately drop all the drugs that you're taking. We would do it by incremental amounts, which itself is not satisfactory, because we need to know which patients are going to progress at different rates so that we can appropriately manage the immunosuppression. I'm not aware that BK interacts with the metabolism of tacrolimus, so I wouldn't expect the presence of BK in itself to increase TAC levels. Although tacrolimus is one of the drugs that suppresses the immune system and can increase the susceptibility to viruses. And then leflunomide, there's a specific question about that. After we have reduced immunosuppression, if there's still a problem with BK virus, we use a number of other agents to try and control it, and one of those is leflunomide. The problem with interpreting its effectiveness is that it's introduced at different times for patients in different situations in regards to BK, and there's a lack of robust clinical trial data, which is understandable in managing an unusual infection. And so the effects of leflunomide are not consistent, and it's one of the agents that's undergoing evaluation as part of the BTS guideline production for the management of BK nephropathy. I've used it. It's in our local protocol as one of the manoeuvres we have to try and control viral replication. I don't know about you in Leeds, Matt. Are you using leflunomide?
So we wrote a protocol about 10 years ago, and in that protocol, leflunomide is part of it, and we're in the process of revising it and removing leflunomide from it. So the data has gradually evolved over time, and some of the early data did seem to show a good effect. As you say, it's very conflicted and very difficult data to interpret. Anyone who's reducing immunosuppression then using leflunomide is doing more than one thing for the patient. So which thing is making things better can be really hard to interpret. Some of the more recent data is a lot less supportive of leflunomide, and I think that's why the Leeds team have felt, well, we're just adding something that might have other side effects. Leflunomide can affect your liver, for example, and maybe if we don't have good data that it's doing anything, we should be looking elsewhere.
And I think this is why it's so important that we're focusing on the development of new treatments. And I think the multi-target attack is very attractive. Because it may be that a small reduction in immunosuppression together with a specific agent against the virus together are enough to control the viral replication and any damage that's being done, whereas either on their own is not going to be enough. So I think this multi-target approach is very attractive.
That's great. Well, we've got such a lovely community of BK researchers in front of us on the presentation today, because if you think about the virus existing in everyone already, that virus has then got to get out of a cell that it lives in, cause mischief, and maybe get into some new ones. So you've got multiple different bits of process there that are happening in the transplant kidney, and our researchers have talked us through all the different parts in the pathway that you could link different agents up. I think, Colin, you might have used the term combination therapy. You can link different agents up to attack all those processes at once. And as you're saying, Sean, combine that with a judicious reduction in immunosuppression to try to get a better outcome in this situation. Sean, it might be good if we could address some of the questions that we're getting clinically about the future for someone who's had BK. I think we've got a question about what happens about retransplantation, and we've also got another question about how appropriate, what appropriate frequency of retesting should be. Would you like to comment on either of those?
I'm sorry, I can't see the questions.
No problem. I can be more specific if you'd like.
Sorry, they have come in. Sorry.
We've got someone whose transplant failed and is asking us really, wants to get back on the list. They're waiting for the BK levels to come down. Once they're low enough and that person has a transplant, do they expect the BK level to come up again? What will the transplant team be thinking about their immunosuppression? So it's really the retransplantation question.
Yeah, and again, that's very variable between patients, and I think we need a better way of risk assessing patients coming into their first transplant and also a subsequent transplant as to what is their risk of it happening the first time or coming back if it's happened once. Generally, retransplantation after the first or a previous kidney has failed with BK is usually successful. Whether that's because when your immunosuppression is reduced, you are able to mount a more robust response, which has some durability when immunosuppression is reintroduced. I'm not sure. Certainly, we've had patients locally that we've reduced the immunosuppression, the BK has disappeared, and we've then been able to cautiously increase immunosuppression again without it coming back. But I note the comment that from another patient that BK rises as soon as he restarts mycophenolate. So the balance is different for individual patients. And then if BK is undetectable, I think whilst your immunosuppression regimen remains stable, you probably don't need to have surveillance testing for it. However, I think if your immunosuppression is changed or increased again, you may need to have further surveillance. So again, reflecting on our local experience, patients we have had who have cleared it on minimal immunosuppression, and we've then wanted to augment that to prevent rejection, we then go back to checking BK, and we do it 4 weekly when they first have an increase for 3 or 4 months, and if there's no change, then we discontinue.
That sounds very similar actually to what we would do in Leeds. It might be worth seeing if we can draw our scientific colleagues into one of the questions, Sean. I think there's one question here that I'd love to know what they think of. It's a question from Fez, who kindly, I think, was the person providing our video. So thank you very much, Fez. Delighted to have you here with us this evening as well as your other fellow patients. Fez has asked, can BK virus ever be fully removed from the system? And I wonder if at a sort of molecular scientific level, Simon, Colin or Andrew would fancy saying what they think about that.
I don't think it could, to be honest. I've never seen any evidence that you could get rid of it completely. I think we don't really know exactly where it hides in the kidneys, or if it indeed hides anywhere else in the body as well. It might do. It's a difficult thing to study because you can't analyze every bit of a person who has BK virus. But I think the fact that it appears to persist for the life of patients, and it looks like people are shedding it, every now and then all throughout life and never knowing about it. I'd be surprised if it can't get cleared by the normal immune response, then I think it'd be hard to ever get rid of it completely.
Yeah, I would agree with Colin. I mean, that there is some evidence that the virus hides in certain immune cells, and so can traffic around the body. But yeah, if healthy individuals who are exposed to the virus cannot remove it completely, I think it would be very, very difficult to do that.
I mean, one interesting comment on that is that there's lots of these similar viruses in this family called polyomaviruses. And there's, I think, correct me if I'm wrong, Andrew, but 13 or 14 human polyomaviruses that live in different places in the body. Some on the skin, maybe some in the lungs. And I think they've been evolving with us so many hundreds of millions of years that they're perfectly designed to live on us forever and not cause much damage until your immune system's a bit out of whack.
Yeah. In fact, actually, one of the problems with this family of viruses is that they're usually very, very difficult to study in the lab. So we are fortunate for BK that at least we can grow it, and at least we have model systems, and hopefully with the model systems that Colin has spoken about, these organoids will have even more information. But there are these other related viruses, distant cousins of BK, that currently we cannot grow. And so we only know about their existence because people have used these very new technologies to look at people's skins, blood, and other tissues and identify viruses, so that they are exquisitely evolved to exist in certain niches within the body.
So taking that question then further, so if we don't think we will anticipate the ability to eradicate, and actually we wouldn't expect that from the biological evolutionary perspective that you're outlining, can that then mean that you have BK as a transplant patient, you have an undetectable level, and then things resurge later? I imagine the answer to that is evident from what we've already said. The answer would be yes. But perhaps the more interesting question is the one aligned to it that I would pose to Simon, about whether even if the virus has disappeared, can it have long-term effects?
Well, yeah, I think you're raising some really tricky things to respond to. But
I'm sorry.
Yeah, we've got probably all got BK virus and JC virus hiding in our kidneys, and then they can become reactivated during certain times of immune limitation. So you would have this kind of waxing and waning infection your whole life, probably. It's interesting because JC virus we actually detect more frequently in the urine than BK virus, but it doesn't seem to cause any urological or even renal problems particularly. We associate it with problems in the spine and in the brain. So the viruses have a lot of biology that we don't understand yet. But yes, the work that we're doing in bladder cells tends to suggest that the DNA damage that's caused in fighting these infections will persist after the infection, and that is a risk in the bladder, at least, for a later cancer development. Now, one thing we don't really understand is whether there's a risk of BK infection or JC infection to renal cells for cancer. And that might be about how the different tissues use apobex. These are enzymes that damage the viral DNA, how they use those apobex to fight infection. It may be that renal cells take a different approach, and we don't know the answer to that yet. So that's something we're going to look into in the future.
Thank you very much, Simon. That's great. There are a couple of other questions in the chat that we could end with. I think we're a couple of minutes before the planned end of our session, so we'll just deal with the last couple of things I think that have come up. Stella has asked, are there any immunosuppressants that seem to prevent? Well, we have, I think, as a transplant community worldwide, researched whether any particular immunosuppression regime is or is not associated with increases in BK virus. There is some evidence that some of the things that transplant doctors give at the time of transplant, the more powerful agents used when it's difficult to overcome someone's immune system, can be associated with more BK virus, but that's not the same as the drugs that the patient takes after they've got the transplant in place. And if those doctors are using those medicines, it's usually because they have reason to believe that they're necessary and are deliberately taking some increased risk in order to achieve transplantation for that patient. But in turning that question back around to the way that Stella has asked the question, is there anything that's simply preventative in terms of the medicines that you as a transplant patient might take for immunosuppression? Sadly, the answer to that appears to be no. Sean, I wonder if you'd like to take the final question for us. Douglas has said that he had BK virus show up when he also had high levels of CMV, and the CMV hung around for 18 months, but the BK subsided, and he's asking, is that the normal sort of thing that happens?
No, now this is a very interesting phenomenon as well, which I think demonstrates the fact that it's different parts of the immune system that are controlling the different viruses. So overall, it's related to a lowered immunity when you're taking immunosuppression, but the specific parts of the immune system may give differential control of the two viruses. I think it's interesting that it's that way round, because I would usually expect CMV, a virus for which we have much more effective antiviral agents, to come under control quite quickly, whereas BK clearance takes a lot longer. So it's interesting that you were that way round. And in terms of immunosuppressants with antiviral effects, this was one of the hopes for leflunomide, that we would be able to use it in that way. Also, cyclosporin seems to be less of a risk factor for the development of BK compared to tacrolimus. These are both drugs in the same class that are used as part of the treatment regimen. But that might be because cyclosporin is itself a less effective immunosuppressant compared to tacrolimus. So it may be related to the drug rather than an antiviral effect. So yes, lots for us still to do.
At the risk of slightly overrunning, Sean, and just to finish your point, would you ever change someone from tacrolimus to cyclosporin on the basis of their BK situation, or is that not a therapeutic move that you use?
Yeah, we have.
Interesting.
We have. So when we have patients with grumbling BK, so it's lower than it was, but not completely cleared in terms of the blood detection. We've used that, and then once we have cleared it, we've switched back to tacrolimus.
Without it resurging?
Yeah.
Fantastic. Interesting. Thank you.
Yeah, so we do have some experience of that. And
So
I think this is one of the things that we're going to gather with the BK rare disease group.
Yes.
So collecting information on patients through radar, we'll know a bit more about the effectiveness of these different treatment options beyond anecdote. So I think it's going to be very helpful in improving our overview of the current situation.
That's great. We're just a minute over our scheduled time, so I'm going to bring things to a close. I'd like to thank everyone. Thanks to Sean as my co-host. Thank you to Simon, Andrew and Colin for their excellent presentations this evening. And thank you very much, everyone, for coming. We're really delighted to have our patients engaging with this topic. We're really keen to keep you engaged, and we're going to email everyone who's registered, as I think I mentioned earlier, with several opportunities that are present for you. One of them is the one Sean's just mentioned. There is a national collection of data around BK. It's referred to as BK Radar through the UK Kidney Association. That's part of the Rare Diseases group within the UK Kidney Association. And in the email that we'll send, we'll mention that it would be great for anyone who wants to, who's had BK, to sign up to that data collection. The second thing is what Simon mentioned earlier about the fact that there is a developing national patient information leaflet here, and it would be really good to have patient commentary on that, and we can put you in touch with Simon after this event to comment on that if you would like to. And thirdly, this evening was meant to be a first step in giving information to patients like yourselves around research. But it's really important that we involve our patients in the development of the direction of travel of research in BK virus, as in all areas of research. So we want to offer people the opportunity to come to future smaller group events where we'd like to hear more from you rather than from us about your experience and about your thoughts about research and research directions around BK. So those three things will come out on email to everyone who's registered, and you'll be able to reply and get involved if you would like to. So thank you very much, everyone, again for our presentations and for coming this evening, and we'll draw the meeting to a close. Thank you very much.
Thank you.